All right, so welcome everyone. Thank you for joining us tonight for our second Better Together seminar series. We're featuring tonight Professor Lapisco Encinas from the Rochester Institute of Technology. We are the Diversity, Equity, Inclusion, and Respect Committee of the Rochester section of the American Chemical Society. And one of the first things that we did when we first became a committee was to put together a diversity statement. This diversity statement highlights our local section core values and embraces our welcoming attitude towards improving diversity at all levels of our organization. In 2020, we were awarded a DEIR grant, a grant from National ACS that is targeted to fulfill the following goals. The first goal is to build awareness around the broad array of career opportunities in chemistry available to students. This is basically planned to be done through quarterly seminars featuring local scientists and engineers' work, where the attendees can learn from the achievements and experiences of this chemist and be inspired. We also want to promote representation by providing opportunities to local, underrepresented, deaf and hard of hearing, and international chemistry and chemistry interested student communities. And we also want to provide a platform to get advice on establishing and maintaining a productive professional career. We want to do this through the Q&A session for discussing how to surpass the challenges, boundaries throughout early career development between the questions that the participants have and the answers that the featured speaker can give. And we also want to do some networking for strengthening the support networks of our student communities and enable them to interact with professionals and other peers. The agenda for tonight involves the speaker presentation, followed by the Q&A session moderated by James Astaire. Then we will have some networking opportunities through breakout rooms, and we'll wrap up with some closing remarks and announcements of future events. All right, so tonight we have the honor to have Dr. Blanca Lapisco Encinas with us. She is a professor and a founding faculty at the Department of Biomedical Engineering at the Rochester Institute of Technology. Dr. Lapisco Encinas received her bachelor's and master's degree in chemical engineering from the Instituto Tecnológico de Sonora in Mexico, and her PhD also in chemical engineering from the University of Cincinnati. Prior to joining RIT, she was a postdoctoral researcher at Sandia National Laboratories and held positions at Tennessee Tech in the US and Tecnológico de Monterrey and Cinvestav Monterrey in Mexico. Her current research efforts are focused on the development of electrokinetic-based microdevices that would answer the needs of a wide array of applications, from cell assessments for clinical biomedical applications to food safety and environmental monitoring. Her research work has been funded by the NSF and other funding agencies in the US and Mexico. And she has received research awards from the Mexican Academy of Sciences and the L'Oreal for Women in Science program. More recently, she received the 2020 Board of Trustees Scholarship Award at the Rochester Institute of Technology. She also serves as editor-in-chief for the journal Electrophoresis, published by Wiley, and has served as vice president and counselor for the AES Electrophoresis Society. She has published over 130 peer-reviewed research articles, conference proceedings, and book chapters. Dr. Lapisco Encinas is a strong supporter of undergraduate research, and many undergraduate students working under her guidance have published peer-reviewed journal articles, including first author publications. That's amazing. And with that, I will pass it over to Dr. Lapisco Encinas with her talk entitled Rapid Bioparticle Analysis with Electrokinetic Microdevices.
Oh, thank you so much, Carla, for such a wonderful introduction. Thank you so much. Let me go ahead and share my screen. And here we have my presentation. So thanks again for this invitation. It's a great honor to be here. Okay. So I wanted to start, I didn't know that your introduction was going to be so great. I wanted to start with a very brief bio, so I'll go quickly. I graduated, I'm from Mexico, right here in the north of Mexico. This is the state of Sonora. And my bachelor university, my alma mater is Instituto Tecnológico de Sonora, right here, very close to the beach. And this is the city of Ciudad Obregón, Sonora, and the nearest beach is only 50 minutes away. Very nice. Then for my master's degree, I actually moved right here to Central Mexico to Instituto Tecnológico de Celaya, right here. Very different type of weather. People talk slightly different there. There's a different accent. I'm from the north. We are supposed to be very loud people. So over here, I learned about a lot about my own country from Tec de Celaya. Later, I came to the United States and I went over here to University of Cincinnati. And Cincinnati, Ohio is right there at the border between Ohio and Kentucky. So very nice area. And after that, in my PhD, that's where actually when I became interested in microfluidics. I worked during my PhD on the development of a microchromatograph. After that, I got an amazing opportunity all the way right here in California at Sandia National Lab, where I continue working with microfluidics, but in this case was a new area. They introduced me to electrokinetics. That is the application of electric field in those little microfluidic devices. And it was like opening a new world, a new field for me. After that, as Carla mentioned, I have held several positions in Mexico and the United States. And right now, I'm a professor and founding faculty of the Biomedical Engineering Department at RIT. We started the department back in 2012 with 3 faculty members. Now we are around 12 faculty members and we are a more established department. But 9 years ago, it was a tiny department. And we are housed in this building. It's called Institute Hall, and we are right here on the 3rd floor. Maybe you can see here, this is the hallway, and in that hallway is where we have our laboratory, and we have very nice laboratory with big windows. It's a pleasure to work there. Okay. So before I get started, I wanted to say thank you to the people that actually do the work. You have here in front all my students. Majority of them are at RIT, are undergraduate. Nicole, she just graduated. She's a PhD student. But Adriana, Cody, Abby, and Kel, undergraduate students, and all of them had published their work in very nice journal. I also have a very nice collaborator, Dr. Julie Thomas from RIT, and we have Roberto Braulio and Victor and Braulio, and those are collaborators from Mexico. In particular, Roberto and Victor are my former students, and they are now professors at the Tecnológico de Monterrey in Mexico. So it's very nice to see that my former students, my former PhD students have now faculty positions. Okay, starting a little bit with the actual talk, what is microfluidics? Microfluidics, if we go to the definition, is the manipulation of tiny volumes or tiny amounts of liquid in little devices. Why people are interested in microscale or working microfluidics? Because with microfluidics, we have the potential of have devices that are gonna give us the result of an analysis in a very short time. Devices that are portable, so you can take the laboratory where it's needed. And microfluidics, as we have in the slide, has revolutionized the separation science because now we can do the separation much faster in portable devices and also in devices that are perhaps inexpensive. A big issue, for example, when we are talking about clinical applications, is that sometimes people don't have access to analysis because analyses are too expensive. So if we can build microfluidic devices, we can create point of care devices that are at low cost, making healthcare available to everybody. Electrokinetics is one of the main pillars of microfluidics. I want to be very clear here, microfluidics is a very big field. We have from acoustics microfluidics to pressure-driven microfluidics. Electrokinetics, which refers to microfluidic devices that employ electric field, is one of the main pillars. Why? Because they're very easy to manipulate. In particular, electrokinetics, we are using electricity, electric field. So we are relying on a physical phenomena. We don't require any chemical reaction. So this makes our processes and devices quite robust because we just rely on physics. And in particular, I want to talk to you about today about nonlinear electrokinetic effect. And I will be explaining what does nonlinear mean a little bit later. But what is interesting about nonlinear is that nonlinear electrokinetic effects can have highly discriminatory capabilities. That means that, for example, if I need to do an analysis to differentiate between normal E. coli cells and perhaps antibiotic resistant E. coli cells that are super similar, if I look at them in the microscope, I cannot tell one cell from the other, but perhaps the antibiotic resistant cell has a different proteins on the surface that will produce a different electrical charge on the cell. And I might be able to discriminate between the two types of cells just by noticing that little difference in electrical charge. So that is non-electric microfluidics. It's like a super weapon. And I might be able to distinguish between cells that otherwise seem very similar. Okay. Nonlinear electrokinetic effects can offer to us high resolution and high selectivity, selectivity being key. And here I have a representation of one of the channels, for example, that we use in my laboratory. They're very simple devices. As you can see, it has an inlet reservoir, an outer reservoir, and there is something in the middle of the channel. We call those insulating posts. And they're pretty much like columns made of the same material of the channel. The channel can be made of glass or polymeric material like PDMS, polydimethylsiloxane. And what is that is these devices are inexpensive. The whole device is made of a single substrate and the electrodes are external. So I use the device, and because it only costs a few cents of a dollar, I can throw it away and then I can do another analysis, and these devices can be disposable. And here I have a representation. There was actually one of my first student in Mexico. He did his master with me. And here we have a representation of a channel that he used. We actually took some water from a pond that we have in the university campus. And after we filter the water, we spike it with E. coli cell and yeast cell. And here is a representation. The red circles right here represent the Saccharomyces or yeast cell, and the green circles represent the E. coli cell. He put that into a device, he applied his magic with the electric field, and what I have here is the actual Excel plot. We just added the color for drama. And if we look at the x-axis, it's 110 seconds. He was able to separate the E. coli and the yeast cells in less than 2 minutes. And in less than 2 minutes, he actually performed 2 different activities there. He separated the cell, as you can see, the peaks, similar like chromatography, and he enriched them. We can see, for example, the E. coli. Here I have the concentration over the initial concentration. He enriched those things 50 times. And for the Saccharomyces, 34 times. So he did simultaneously separation and enrichment in less than 2 minutes. If we wanted to do something like this, separation and enrichment, like with microbiological techniques, normal techniques, instead of 2 minutes, you're talking about 2 days or perhaps 2 weeks, depending on what type of processes you're talking. So 2 minutes, master student, you can see here the representation. Okay. Now, what is this techniques that I'm talking? Insulator-based electrokinetic. It works like this. Here I have a representation of the channel. You can see a 3D representation, and then I have a top view. You can see I have an inner reservoir. This little blue thing is like my little electrode. I use platinum wires. This is the inlet, and this is the outlet. And these circles in the middle, I call them the insulating structure. Here in this representation, I have circular ones, but we make ones diamond shape. We even have used some that we call asymmetric ones, in which one side of the post has one shape and the other side has a different shape. So we play with these geometries a lot. But here I'm showing you the simplest design, just the circular one. Now, how are these posts? Imagine these posts like the column that are holding the roof of a house. So if I'm inside the microchannel, imagine this room is the microchannel. These round columns go all the way from the floor to the ceiling. So they can create a true three-dimensional effect. And what is happening is the following. Here, these red lines represent the electric field. We can see, for example, in this region that the electric field is very nice and homogeneous. But once the electric field gets to this region that we call the insulating poles, the poles are insulating because they do not conduct electricity. They are made of a material that is a bad conductor. So the electric field is not able to penetrate the poles, and therefore it has to squeeze in between the poles in these regions. And that creates in this region, which I refer to as the constriction between the poles, what we create there is an area where the electric field is more intense. In this way, we are creating, and it sounds like super fancy, a non-uniform distribution of the electric field. The electric field is uniform here, but then it is highly concentrated or higher magnitude, lower magnitude, higher magnitude, lower magnitude. Now, why do I want that? For a very specific reason. Those nonlinear electrokinetic effects that I mentioned that can have very high discriminatory capabilities, they are only awakened at high electric field magnitudes. So I'm putting those little traps in there in order to create specific regions when the electric field will have a high magnitude. So those nonlinear effects that I want are going to become active here, but only here. In that way, I can control where exactly I want the forces to be applied. So, and one thing, these channels are like super simple. The whole thing is made of PDMS, the roof, the floor, and the structure. So one single substrate, super easy to fabricate. My undergraduate students that work so hard, it takes them maybe 90 minutes to make a channel. So very nice to make. Okay, now I want to talk very briefly. I know that a lot of people here have a background in chemistry, so perhaps just a brief introduction of what are the phenomena that we are playing with in these insulator-based electrokinetic devices, okay? So the first phenomena is electrophoresis. I think 100% of the people present here are familiar with electrophoresis. So let's go quickly. Is the movement of particles because they have a charge with respect to the liquid. Electrophoretic is mainly movement to a Coulombic force. And one characteristic that is very nice is, in particular for bioseparation, majority of biological particles have a charge. For example, DNA, because of the phosphate backbone, has a negative charge. Proteins, majority of proteins like pH above 5, they're going to have a negative charge. And I can tell you that we have also measured in my laboratory the charge of many bacterial and yeast cells, and everybody's negative. So many biological particles have a charge, and by the way, that charge happens to be negative. So we can use electrophoresis to separate them and analyze them. Now, electrophoresis, we have a simple representation here. The particle will move towards the electrode that has an opposite charge, and it depends on the charge to mass ratio. So here in my simple representation, you have a small particle with a big charge. It's going to move very fast. And then a larger particle with less charge is going to move slowly. A particle with no charge is not going to move due to electrophoretic. And particle with the opposite charge is going to be moving in the opposite direction. Now, another phenomena that I would like to talk about is electrosmotic flow. I remember when I went to Sandia Labs, when they first taught me electrokinetic, I couldn't believe it. I remember them explaining to me, Blanca, you're going to do these experiments and you're going to put liquid, and the liquid is going to move from the inlet to the outlet of the microchannel. And I remember looking at the channel, and it's like, trained as a chemical engineer, that's fantastic, but where is the pump? How am I pumping the liquid from the inlet to the outlet? And they told me, oh, don't worry, just use electrosmotic flow. No one had told me what electrosmotic flow was, but it's a fantastic flow liquid mechanism to use in microfluidics. Why? Because when you are creating a microfluidic device, you want to create a really miniaturized device and not a chip in a lab. That you have the chip, this is small, but it's connected to a lot of really huge equipment. That is not what we want with microfluidics. I want everything to be miniaturized. So if I don't have to use a micro pump, that is making my device a little bit more robust and more miniaturized. So let me explain how electrosmotic flow works. Many materials, glass, PDMS, PMMA, Another material used in microfluidics, they have a wonderful characteristic. When we put those materials in contact with a solution that has ions, the material by itself, for free, that's how I explained it in my microfluidic class, acquires a charge. So imagine my hand is the material. It's in contact with a solution, the material will acquire a charge. Another characteristic, all the materials that I have worked with in my laboratory, they happen to achieve a negative charge. So if I have ions floating, if the surface has a negative charge, positive ions are going to be attracted to that charge. So let's look here in the slide. I have a very simple representation of glass. Glass has a negative charge, then a bunch of positive ions are going to be attracted to the surface. Now let's think that we are in a microchannel. Imagine, for example, the room that you are in. If you're in a microchannel, the floor, the ceiling, and the sidewalls are going to have all that charge. So imagine the ions being attracted to all those surfaces, okay? So you have the negative charge, you have some positive ions. Then because now you have a large concentration of positive ions, some negative ions are also going to be attracted to kind of balance the charge. That is called the electrical double layer. It's called the double layer because those ions that are on top of the surface, some are going to be in a layer that is called the compact layer and then the diffuse layer. This is a very simple explanation, but why is this relevant? Let me tell you. Imagine a microchannel. Here I put my positive electrode and here the negative electrode. And as I mentioned, since the surface is negative, the ions that are floating here are positive. So when I apply an electric field, all those positive ions, so they are attractive to the negative electrode, and as they move, they also move the liquid with them. So something very interesting about electrosmotic flow is movement of the liquid that starts from the walls to the center of the channel. Completely opposite from hydrodynamic flow, or also called pressure-driven flow. Anybody that has taken a fluid mechanics class, we know that hydrodynamic flow has the highest velocity right in the middle of the channel, and the velocity at the walls is zero. Well, here, the movement is actually starting at the walls. A very important characteristic to mention about electrosmotic flow, only works in microfluidics, is unique to microfluidics. Why? Because you need to have sufficient surface area to move the volume of liquid inside the channel. If you have a big channel, your surface area will not have enough. A lot of microfluidics rely on surface effect. And a very important characteristic of microfluidics is the surface to volume ratio are quite high. So unique to microfluidics. So you apply an electric field. I remember the first time doing this, and yeah, the liquid moves. You can see how the liquid start moving to the outlet. And that's how it works. It's just, we are moving ions and the ions move the liquid. So here we have it. This is a video. You should start playing in a second. A video of electrosmotic flow. For this particular video, I put some green particles. Let me see why it's not saying. Maybe needed another click. Give me just 1 second. Okay. Let's wait just for a second. Let me just go back into the presentation mode. I tested just before, so. Okay, it is playing now. Okay, we are in business. Okay, so I put a couple of, oh, come on. I put a couple of green particles so we can actually see the movement of the liquid. Maybe it wants to be here. Let me see if I can play it over here. I'm just waiting for a second. I have the 2 monitors, and maybe the 2 monitors is confusing my computer. So since it's playing, I will do nothing. So we have some green particles, and as you can see, the speed at which the particles are moving depends on how much potential I'm applying. 500, 800, can you see that they're moving faster? Now, what happens if I apply no potential 0? What happens if I flip the polarity? It will just go backwards. So something that electrosmotic flow will give you can switch direction very easily. Very simple, you just change the polarity of the electrode, you can send the flow backwards. Something that will be impossible to do with a pump. I mean, to make a pump go backwards, it doesn't happen that easy. And I'm trained as a chemical engineer, so this is a very nice phenomenon. And actually, in my laboratory, we did a refinement of a method for measuring electrosmotic flow. One characteristic of our system is for our systems to work, we need to have liquid inside the channel that has a very low concentration of ions. Why? Very simple. We are applying a current, an electric current, into a very small channel. If we have a lot of ions in the liquid will conduct too much electricity, and too much electricity will produce heating. Joule heating, that's how it's called. And that can also produce electrolysis, and the heating itself is very undesirable effect, because if I'm separating proteins, which we have done in my laboratory, you will denature the proteins with the heat. And if I'm analyzing cells, in particular G cells, for example, which are eukaryotes, they tend to be quite fragile, or mammalian cells, a little bit of heat and I will kill them. So I don't want the heat because it denatures proteins and it can also kill cells. So I'm forced to have suspended media or liquid that have very low conductivity. So we had to develop our own methodology for measuring electrosmotic flow in systems with very low conductivity. And this is the paper that we published on that. It was a refinement. We didn't invent current monitoring. It's a technique that exists for a long time, but we just developed a refined methodology for us. Okay. Now, I wanted also to mention another course. This is called electrophoresis. And for a very long time, it was thought that was a dominant effect. I will explain a little bit much later. But let me tell you what it is. The electrophoresis is the movement of particles due to polarization effects. That is not because the particle has a charge. The electrophoresis to happen has one requirement. The particles have to be exposed to an electric field that is non-uniform. That is an electric field that is stronger on one side and weaker on the other side. Now let's look at my little cartoon over here. I have here, can you guys see my mouse, or you don't see the pointer? This is mouse? Perfect. I have here a larger electrode and a smaller electrode. And by having that configuration, the electric field closer to the smaller electrode is stronger. Now let's look at the particle. In theory, this particle is neutral because it has 3 positive charges and 3 negative charges. However, because the positive side of the particle is exposed to an electric field that is stronger, the particle will get polarized. And the charges in the particle are unbalanced. Why? Because the electric field is not uniform. And that actually creates particle migration and particle movement. One advantage that electrophoresis has is the electrophoresis doesn't care if the particle is neutral or the particle have charge. The electrophoresis will work. So in that case, the electrophoresis is more flexible than electrophoresis. Electrophoresis needs the particle to have a charge for electrophoresis to work. Another thing is that the electrophoresis can work with both DC or AC electric field because it doesn't care about the direction of the field. The only thing that the electrophoresis needs is the field to be stronger in one region and weaker in another, and that will work. And this is one of the nonlinear effects. And don't worry, I'll be explaining what is this nonlinear thing that I keep talking about. Okay. So now that we have explained very briefly electrophoresis, electrosmotic flow, and the electrophoresis, I wanted to show you this little cartoon representation of the forces that we have. This is a microchannel, and this is actually built from dimension. I got the image from our AutoCAD modeling. This is how our channels look. And in this case, I have cylindrical insulating posts. You might be wondering why the first post have like a cone shape. And that, I would like to tell you, it was because the mathematical model suggested that. No, this was just a practical application. When we have normal posts that were round, and we have, for example, cells, and we apply the electric field, the cell used to go and just crash themselves against the post and splatter, and just create a big clog and clog the system. So by putting that conical shape, the cells do this, and the system is no longer clogged. Sometimes practicality has to be included in this design. Okay, so here I have the representation of 2 posts from here. and the red dot represents a particle. We have electrosmotic flow that is going from left to right. Why? Remember that this channel has a negative charge. So the ion floatants are positive. So those positive ions are attracted to the negative electron. So electrosmotic flow is moving to the right. Okay. Also, we have electrophoresis. Can you see that I have two of those? I have the linear electrophoresis, which I put the little one just to state that it has a linear dependence with electric field. And I also have that electrophoresis of the second kind. I know it sounds like something from outer space, but it's of the second kind and it has a 3. Why it has the 3? Because it has a cubic dependence on the electric field. So I will explain that, but we have two different types of that electrophoresis, and then we have the electrophoresis, and the particle is feeling all these forces. And here I have another representation that we can use these forces to trap or capture particles. The image on top is a microchannel, and I just drew how trapped particles look in the microchannel. How can we trap the particles? Particles are trapped when their velocity becomes 0. How can I do that? Is when all the forces exerted on the particle are in equilibrium. If everybody pushing this way and everybody pushing the way are pushing at the same magnitude, the particle is not gonna move. So in this case, I have electrosmotic flow, which is going from left to right. And then we have the two electrophoretic forces and the electrophoresis pushing in this direction. And when they are balanced in the location, when the forces are balanced, that's when the particles are trapped. And that trapping can be quite substantial and is fantastic for enriching particles. We know that a big challenge in bioseparation sometimes is that the samples that we get are very dilute. You know, I wanna do the analysis. I know that the particles I need to analyze are there, but the concentration is so low that I cannot do the analysis. So if a little device like this can concentrate your sample and with that enable to do further analysis, that's already a big win because concentration is a big issue. So here we have the forces, and to achieve particle trapping, we have electrosmosis in this direction and two electrophoresis in the electrophoresis in the opposite. Okay. So I also wanted to mention what are the regimes in migration regimes in this system. We have two migration regimes of type of movement that the particle do. One is called streaming, and is when you have linear and non-linear forces working together and the particle move in streamlines. You will see that happen when the electric field is not high enough for the non-linear effect to be dominant. If you remember, I mentioned that non-linear effects are super cool and they are very effective, but they have one characteristic. They require the electric field to be high enough for them to be active. So when you have an electric field that is not high enough, non-linear effects are present, but they do not dominate. And they will do something like a streaming, which is what I have here when we apply a potential of only 200 volts in a channel like this. Then, when I increase the potential to 600 volts, we actually get trapping. Very similar to the drawing. This is the trapping right here. Hopefully you can see the post and the trapping. And here, for example, we are looking at 12 traps, and it's just so beautiful how all the different traps are equal to each other, okay? With this, I think I have explained you what are the forces acting. So maybe, and hopefully it's gonna play, yeah. There is a video of one of these systems. These are 1 micron particle, the gap is 50 micron, and please see what is happening each time I increase the volt. Can you see the particles jumping and jumping? Because each time I increase the electric field, the equilibrium location changes position. That's, for example, what is happening. So this is linear electrokinetics. The particles are just flowing. This is what we will call streaming with trapping. And as I keep increasing and increasing the electric field, we go into full trapping. The highest potential is 800 volts per centimeter, and that right there, that's trapping. And I want you to see how far this is from the center of the channel. And something that is very important in these effects, when it's electrokinetics, it should be reversible. The moment you remove the electric field, poof, the effect is gone. And you see that at the very end of the video. I'll wait for this to play one more time. After I reach 800, I remove the electric field, and it's because it's through electrokinetic. Oh, well. After that introduction, I would like to talk to you about something very new. Actually, this concept of the electrokinetic equilibrium condition, we discovered that last year. These are our work from 2020. Somehow the pandemic worked for us and we were able to do this. Okay, so let me just give you a little bit of some history about the electrophoresis. When we have all these systems, we used to call this system that I now call insulator-based electrokinetic. We used to call it insulator-based electrophoresis because we thought, we were not correct, that electrophoresis was the dominant effect. And we noticed something. Many people like myself, half of my research is mathematical modeling. We do modeling with COMSOL and MATLAB, and we predict the performance of the system, and we use the Laplace equation and boundary conditions and all that fancy stuff. But we noticed something. The results from the model, they never matched the experimental results. And we were scratching our heads, and then we thought, maybe we have Joule heating in the system, maybe we have some electrolysis happening, maybe we have particle and particle interactions, but the truth is, whatever the model was predicted, never matched the experimental results. So people like myself used to add to the model something called the correction factor. Sounds very fancy, but we used to add like a number, multiply the result by something, so it will match the result. And actually in 2019, my student Nicole and I wrote a review paper on the use of correction factors. And here you will see a big table with a lot of the people that use correction factors because we needed, the models were never accurate. Guess what? We were actually missing an effect. We have, as I explained in this system, linear electrokinetics. And I will explain why the linear and non-linear. Linear electrokinetics is linear electrophoresis. As you can see, the velocity of a particle moving and the electrophoresis depends on the mobility and the electric field to the power 1. That's the linear part. When I go to electrosmosis, the equation is very similar. The mobility multiplied by the electric field to the power 1. And you can see this symbol right here is called the particle theta potential, and it measures the charge of the particle in units of millivolts. And for all the particles that I used, they were negative, with negative zeta potential. This right here is the zeta potential, the W is for the wall, because in the same way that we measure the zeta potential, the charge of the particle, we also measure the charge on the walls of the charge, okay? But these two are linear. You see electric field to the power 1. Now we have this electrophoresis of the second kind, and I call it EP3. The 3 is because it has a cubic dependence on the electric field. So once we hit the electric field where this phenomenon is important, if the electric field increases just a little bit, the electrophoretic of the second kind effects increase a lot because it has a cubic dependence on the electric field. And then we have the electrophoresis, which is also nonlinear, even though it depends on the electric field being non-homogeneous. In engineering terms, it's called the gradient of the electric field, but you can see that it has a square dependence. So this is cube, this is square, both of these are nonlinear. Okay, now what's the difference, like the physical difference between those phenomena? Linear effects depend on the charges that are on the particle. For example, the polystyrene particle that I showed you in my video, they are coated with a carboxyl functionalization, and that thing is negative. But it's the charge that is actually on the particle. Many bacterial cells and yeast cells that we analyze, they have proteins in the surface, therefore they have a charge. So linear electrokinetic phenomena depends on the charge that is on the particle that is there. Non-linear electrokinetic phenomena depend on the bulk charge. That is the electrical double layer, the ions, the cloud of ions surrounding the particle. That is the main dependence. Now, when we came up with the concept of electrokinetic equilibrium condition, that only means the condition at which the forces are in equilibrium. And the first thing that we did, we went back to the drawing board. This is a little microchannel with an inlet and an outlet, and here are particles. Do you see that I don't have any insulating post? So little straight channel, just go. In this type of channel, the velocity of the particle will depend on the linear electrophoresis because the particles have charge. Electrophoresis of the second kind because the particles have charge, therefore they're going to have the ions surrounding the particle, and electrosmotic flow. As you can see, I didn't put the dielectrophoresis in here because for dielectrophoresis to happen, I need the electric field to be non-uniform. And in this channel, beautiful and homogeneous, the electric field is homogeneous and in the same. So this is a beautiful homogeneous electric field. There is no dielectrophoresis. Okay. So we did something called particle image velocimetry. Let's look at this plot that we have here. We apply different electric field and we measure the velocity of the particles. We have particles that are 1 micron, another particle that is 1.9, almost 2 micron, and then 5 micron in diameter. And we notice the following, which we have never done. At low electric field, the velocity of the particle increases linearly, which is understood because we have the linear effect. As you can see, for the 3 particles, it goes linear. Then it reaches a maxima, then it decreases, and then it became negative. And we were like, what? The particles are like going backwards? We repeated the experiment with many particles. Each one of these experiments, you can see the error bars. It was on the repetition. So they started moving. Then they arrived to maximum velocity. Then the velocity decreased and then negative. And we were like, what? And we noticed that there was a different value at which each one of the particles crossed the 0 velocity. Then we look back at the equation. We have linear electrophoresis, the non-linear electrophoresis, that's how we call the second kind, and then the electric field. So we simplify the equation, eliminate the electric field, then we have electric field squared remaining here. We can actually calculate it. We figured it out that we can actually calculate the value of this by using these mobilities. It comes from this equation by making velocity 0. And this gave us this electric field value here, this one here, and this one here. And we noticed that it was unique to the particle because something that expression has right here, it doesn't depend on system characteristics. I can have a channel that has pulse, a channel that has no pulse, a channel that has pulse in diamond shape, in oval shape, or whatever. That EC value that we are calling unique electrokinetic signature remains the same. It's a system independent parameter. And we thought, wow, maybe we can actually identify things by this parameter. So the story continues. After we learned that with the microparticles, we did the same thing with different type of bacteria and yeast cell. This is an analytical chemistry paper from 2020. The other one was also from 2020. So we learned that there is that electrokinetic equilibrium condition. We call it ECC. Now, what would happen if we have the channel that I showed you at the beginning, the one with insulating post? Okay, so here is a representation of the channel. You already know this image that I showed you. And if you remember in this channel, I spent quite a bit of time explaining you that there are 4 forces: electrophoresis, electrosmosis, the other electrophoresis, and dielectrophoresis. So here are my 4 forces, but we noticed something. We did some careful experiment with bacteria, and if you look at that little table, dielectrophoresis is tiny. For 20 years, this system, people call them insulator-based dielectrophoretic devices, and we thought that dielectrophoresis was trapping the particles. No, it's actually 2 orders of magnitude lower than all the other phenomena there. So if I write the equation, I can actually remove this. And if I remove it, look at what I'm left with. I'm left with exactly the same forces that I had for the electrokinetic equilibrium condition. So that means that even in devices where I have insulating post, I can use this parameter because the other one, dielectrophoresis, is present. Don't get me wrong there. It's just tiny. It's not making that much of effect. And why will I want to use these insulating devices? I can tell you the reason. You saw in my collaborators, for example, Dr. Julie Thomas, sometimes we have viruses. Those things are tiny. If I want to do velocimetry measurement with viruses, it will be very hard unless I have a fantastic microscope or something because they're very small to see. So trying to do velocimetry tracking on very tiny particles or particles that have a weak fluorescent signal, simply not possible. Something that we came with then was the an application of the electrokinetic equilibrium condition with insulating poles. Let me show you. You saw from my videos that we can trap particles between those poles. And I have showed the forces that are creating the equilibrium. Here, right here, these are E. coli cells that we actually label with a blue fluorescent dye. And I know, and from this picture, that is an experimental picture, I put this little cross, noting what is the location when the band of particles or that agglomeration of particles start. Because that is where I have my electrokinetic barrier created by the electric field that the particles cannot penetrate. Then I go with my fancy COMSOL model and plot the electric field as something called isolines, which are lines of fixed electric field. And I noticed that these locations corresponds to this cross. And this is, for example, something like 1100 volts per centimeter. In that way, from that single image of the particles being trapped, I can extract the electrokinetic equilibrium condition. Since this one is extracted with insulating poles, I just added empirical electrokinetic condition just to acknowledge that a little bit of accuracy is lost because I'm not considering the electrophoresis that can be between 2 to 5%, but it's actually pretty accurate. So let me show you how we used it. And we actually used that with cells. We did a bunch of experiments in a device with cylindrical poles, and you can see I did, what I did is my students that are amazing did E. coli, B. cereus, B. subtilis, a second E. coli, Saccharomyces, and Salmonella enterica, 6 different right here. And we measured what was the voltage required for us to have a nice band of trapping. And you can see the different voltage for different cells. Then from that image, with the process that I explained, we used COMSOL and we estimated the empirical electrokinetic conditions. We used that and we obtained that, and we call that electronic signature. And you can see that we have different values for each cell. And then guess what? We have claimed that this is gonna be system independent because if you look at the expression right here, it doesn't have anything related to the size of the system, the shape of the poles, the gap between the poles, nothing. That only depends on mobilities, and the mobilities are properties of the particle. So what we did, we actually, after we obtained that electrokinetic equilibrium condition, here we estimated what will be the trapping voltage, getting that value for oval-shaped poles, just to see. We got the ECC value that we got from experiment with circular. Let's see what now we can predict for oval-shaped poles. And to make this better, we actually did the experiments. So here are the experiments with the oval, and we measured it. One is the trapping voltage that we predicted with the COMSOL modeling, and the other one is the experimental one. Look, for example, the B. cereus, the results are very close. Look at the B. subtilis, which is the second set of bars. The blue one is the experimental one, and the red one is the predicted one. Can you see that it's almost identical? So for the very first time, we were able to have predictions that actually match the experiments and without using any correction factors. And what we learned here is not that the models that we had were wrong, is that we were not considering a full effect. Now, this phenomenon of electrophoresis of the second kind had been well known by the people in the physics field for quite some time. But somehow that nugget of knowledge never made it to the people like myself, and my field has many chemists, biologists, chemical engineer by myself, biological engineer. Somehow we just never learned this. And by bringing that concept, we now finally have models and experiments that agree. So these are, as you can see, we have very nice agreement between modeling and predictions. And with this, I would like to finish. I know that we run a little over time, but there is great potential of having this electrokinetic condition for identifying cells. Like if I see a cell in a given device and it's trapping at 500 volts, once I have my library of data, ooh, that looks like E. coli. And if it's trapping at 700 volts and I see my data, oh, that looks more like Salmonella. So it could be a way to rapidly identify microorganisms. And there is plenty to learn. I have been working in this field for quite a bit of time, but we keep learning about new phenomena. And finally, I would like to thank again the committee for the invitation and the NSF for the funding. Thank you so much. And I have here, hopefully it's gonna play. I call it the electrokinetic dance. We are using AC signals, so we are changing the polarity, and you can see the cells dancing from one side to the other. Thank you so much for this invitation. Thank you.
Thank you so much, Dr. Blanca. That was amazing.
Oh, thank you so much. And I'm sorry, I know that we still have a couple of activities to do for today. Thank you.
Oh, it's no problem. So does anyone have any questions over the talk?
I don't bite.
I do. Does shape of the particles make a difference? Like anisotropic particles?
Yes, and that is something that we are actually investigating right now. Cells, biological cells like bacteria, yeast are going to allow us to do that better. We usually test our system with polystyrene particles. All of them happen to be spherical, but the shape matters. Shape actually affects the electrophoretic mobility, and for the little influence that electrophoresis will have, the polarization, for example, of a prolate particle or a rod is different from that of a spherical. So yes, shape matters for linear electrophoresis and non-linear. Imagine, for example, I don't know, something like my cell phone, and we have the cloud of ions. That bulk charge for that non-linear electrophoresis depends on is going to have a different shape if the particle is not spherical. Yes, absolutely. Fantastic.
Thank you.
Question. Thank you.
Thank you.
Anyone else?
Any more questions? I'm happy to answer any questions.
I have one. So in your bio, we learned that you have like over 130 publications in many different areas. So how did you how did that happen? How did you make that?
I get really good at students. That could be the recipe. Now, in particular, for example, my lab is not that big. Like I don't have that many students at a given time, but I do work very closely with them. And as I mentioned, a lot of the publications, the numbers can be that big because the undergrads are publishing. RIT, different, for example, from University of Rochester, we do not have such a big graduate program. So the workhorse of my laboratory, the people that make the magic happen, it is a lot of the undergrads. Something, for example, that I have noticed that works really well, when we are working together on a project, we write what we call the plans. And having the objectives of the research very clear kind of empowers people with knowledge, and they can go ahead and do the experiments and make decisions on their experiments. Another thing that I found that is very good is empower people with knowledge. This lecture, for example, that I gave to you, I present similar slides to my students so they understand the phenomena. In that way, when they are doing experiments, the electrosmotic flow is not working well because the particle is going backward or something, they can actually make decisions. I might add the right decisions because they have been empowered with knowledge. So a lot of the papers that are being published is my undergrad students are great. They are working a lot and they're productive.
Awesome.
Does this work on the molecular level, like individual polymer chains?
Yes, we have actually done some earlier studies on proteins and DNA. So people have used this phenomena in proteins and DNA. Yes, because the only characteristic is they need to have some charge, so the electrophoresis is going to move. And by applying the electric field, we're going to have electrosmosis. As long as the forces are present, the phenomena are bound to happen. So yeah.
Let me add on to that. So if I have a particle like a polymer chain, a high molecular weight polymer chain that's charged, and it gets into that shear field between the poles, now you have a high shear. What if it stretches out? How are you going to interpret that? Because in the low shear field, it'll be a sphere, no question. In a high shear field, it'll start elongating. Does it make a difference?
Yes, and I wouldn't want the person to model that with COMSOL. It will be quite complicated. But since we mentioned earlier, all of this depends on the mobilities, the linear electrophoretic mobility and the nonlinear electrophoretic mobility. Those are playing a big role. I'm not counting electrosmotic flow because that doesn't depend on the particle. So yes, if the particle is gonna be shifting shape when it's in the high shear area, those individual mobilities are gonna move. And you know what? No one has done that just yet. It's a fantastic idea. How will act on deformable polymer molecules? I mean, that will be awesome. And whoever is able to model that thing, that will be even more awesome. Mathematical modeling of a shape shifting molecule. That's pretty cool, yeah.
Thank you.
Any other questions?
One question here. Hello. If there is uncertainty with respect to your volt signal, what referee technique would you consider?
I'm sorry, uncertainty with respect to what signal?
This signal for identifying whether you're looking at E. coli or some other type of protein, some specific type of protein. If your reference table for identifying what signal you should be looking at for that E. coli, what referee technique would you turn to confirm?
For example, if it's something, if we are trying to identify and we find a case which is very similar, we have done, for example, when we do different type of bacilli, like Bacillus cereus and Bacillus subtilis, those things are very similar and we get the same signal. So what we do, we might need a phenotype, and for that we might need our collaborators in biology. But from our side, from the engineering side, I might turn to use posts that are a little bit narrower, because those allow me to have higher discrimination. The circulars one that I showed, those are like the workhorse. We get started with those. But we see, we are getting two different types of particles and they are not able to separate each other. I will go with another shape of those insulating posts that are a little bit more discriminatory. That means one particle will locate in one area and the other one is gonna locate in another area. We have seen systems, for example, when we have mixture, we can see 2 or 3 different bands. It's because we have 2 or 3 different types of particles. So we have total control on the shape of those posts. And depending on what is the type of particle that I'm analyzing, I'm gonna do a tailored system. But yes, when we have done several studies on how the shape of the post can give us a more discriminatory system. So that's a very good question. I will play with the geometry of my system as much as I can, and I fully understand we are completely relying on physical effects. There might be some cells that are just way too similar that I might not be able to tell. But if there is a little bit of difference in charge, I know what to do with. So we might be able to do that.
Thank you.
Thank you so much, Dr. Blanca.
Oh, thank you so much for this invitation.
All right, so I think I'm sharing the slides. So just a few of the events coming up. We have our June Rochester section executive meeting. If you would like to come see what events we're working on or want to try to get involved in the American Chemical Society, the executive committee meetings are an excellent opportunity to come meet us and see what kind of projects we have working on. We also wanted to let everyone know that in September, we have our next DEIR event where we'll have a government chemist coming. And in December, we'll have a member from the nonprofit sector as well. And we're open to nominations for speakers. We also are having an open position for member at large for the American Chemical Society's Rochester section. And if you're interested, feel free to email Dick Cobb, whose contact information is here. As another part of the grants, we're also working on making 12 educational videos to post to our YouTube channel. And we are also looking for volunteers for that project as well. If you're interested, feel free to contact me. And that concludes our event for the evening. Thank you so much, everyone, for coming. Thank you, Dr. Blanca, again for your amazing talk.
Thank you so much, James. Thank you.
Yes. And that wraps up the event. Thank you again, everyone, for coming.
Thank you.
Thank you, everyone, for joining.
Nice talk.
Bye.
Gracias, doctora.
Bye.
Yes. Mil gracias. O sea, hablas español porque tu pronunciación perfecta.
Un poquito. Muchas gracias por aceptar la invitación.
Gracias a ti por la invitación. Bye.
Hasta luego. Bye.
Bye. Thank you.
Bye, everyone.